Do you want to get a clear and beautiful view under the microscope? It all starts with preparing a perfect slide. Let's walk through it step by step. For today's experiment, we'll be using slides, cover slips, Tradescantia leaves, forceps, dropper. I have repurposed a medicine dropper for my home lab use, some tissues, water, and of course, microscope. Tradescantia is a great choice for microscopy because its cells are large and store colorful pigments, primarily anthocyanins, which are responsible for the vibrant colors seen in their leaves. This means we can observe them clearly even without staining. I have used a variety of Tradescantia growing in a garden near my home. Can you see the purple color underneath? To prepare your sample, take a fresh Tradescantia leaf and rinse it with water to remove any dirt. Gently tear the leaf using both hands. Place a drop of water on the slide. This keeps the sample hydrated and helps hold the cover slip in place. Look closely at the edge of the tone area of the leaf. You should be able to spot a very thin, almost transparent layer, which is the monolayer of cells. Now use a pair of forceps and carefully lift this thin layer. Remember, no matter what sample you're working with, always aim for the thinnest section possible. Thin samples allow more light to pass through, making it much easier to observe the internal structures clearly under the microscope. Use forceps to place a sample in the center of your slide. I've taken a few small pieces of the purple leaf layer. If the sample is drying up, add another drop of water over the specimen. Now take a cover slip with forceps, touch one edge of the slide and lower it gently at 45 degrees angle. This helps prevent air bubbles from getting trapped. Press the cover slip lightly to spread the water. Wipe away any excess liquid using a small folded piece of tissue paper placed near the cover slip and remove it quickly. Too much of water and your sample might float. Too little and it could dry out. So control the moisture carefully. If the air bubble persists, you may try to remove it gently by placing a cover slip again, or you can just simply ignore it and try to focus in an area of sample away from the bubble. And here you go, microscopic view of Tradescantia cells. Just look at the perfect packaging and vibrant color. They're tightly packed, polygonal, most are purple, some are colorless. I move to the higher magnification and I can clearly see the whole hexagonal cell filled with anthocyanin. Now is the time for a question. Where else do you find such polygonal structures naturally? The answer is, they're found throughout nature in places where efficient space usage is important, like in the hexagonal cubicles made by honeybees. You can catch a housefly in a box, freeze to immobilize it, mount it on a slide and observe under the microscope. You should be able to see the compound eyes with polygonal ommatidia. Or maybe consider watching the Netflix documentary about Kulu's journey and observe the beautiful polygonal pattern of pangolin scales. Often, the hexagonal or irregular polygons minimize gaps and maximize coverage on both flat and curved surfaces. Can you think of any non-living natural structure made of polygons? And do you think all the plant or animal cells will have hexagonal or pentagonal structure? While we think about these questions, let's not forget one last step of the protocol, which is to clean the used slides and cover slips. Greasy or cloudy slides are a big no for microscopy. So, before you wind up, wash your slides and cover slips with some liquid dish soap and wipe them with a clean cotton cloth. Let them air dry. thank you for joining and keep practicing. In the next lesson, we'll learn about how to fix the dirt and blur in microscopic view.